The cell is the basic unit of life that can replicate on its own. The human body alone has over 200 different cell types, from long skinny neurons that can grow over 1 meter long to myocytes or heart muscle cells that contract to let you flex your muscles. But despite their differences, they share many features, including the cytoskeleton. The cytoskeleton is a network of proteins within the cell. The cytoskeleton gives each cell its shape and anchors organelles in place, keeping everything sturdy, kind of like the frame for a house. But it's also a dynamic network, which can change shape when the cell wants to move, contract, divide, or pull in or push out molecules. Imagine if your house could do that. Maybe it'd get up and walk away during an earthquake. So the cytoskeleton is pretty special, and it's made up of 3 proteins: actin filaments, intermediate filaments, and microtubules. Actin filaments are the thinnest of the 3 proteins, so they're also called microfilaments. They're made up of 2 strands of actin proteins arranged in a long twisting chain like a twisted necklace. The actin filaments connect to one another to form a network, like a spider's web, that's located just below the cell membrane. The actin filaments slide closer together and further apart, allowing the cell to change shape during muscle contraction. Not surprisingly, the muscle cells have plenty of actin, as well as another protein called myosin. Myosin filaments bind to actin filaments, and that's what allows the actin to slide closer together and further apart. And ultimately, that makes the muscle cells shrink and stretch during muscle contraction and relaxation. Similarly, sometimes these networks change their shape, and that allows cells to move. White blood cells, like neutrophils, use extensions called pseudopodia, or false feet, to crawl in and out of blood vessels, a process called diapedesis. The way that works is that the neutrophil's actin filaments grow rapidly through the polymerization of many actin monomers in one direction, to push out the cell membrane and create a foot. This newly created foot wedges between the endothelial cells that make up the blood vessels. The neutrophil then begins to squeeze through, until it reaches the other side. It's kind of like squeezing between 2 fence poles to sneak into an amusement park, rather than paying admission. Not saying that you should do that. Anyway, actin filaments also play a role in mitosis, or cell division. At the end of mitosis, the cell has 2 sets of chromosomes that each sit in their own nucleus. A ring of actin filaments then forms around the center of the cell between the 2 nuclei. This ring uses the sliding movement of actin and myosin to help constrict or squeeze the cell so that it pinches off into 2 new cells. Next are the microtubules, which are the thickest and largest of the protein structures in the cytoskeleton. Microtubules are made of alternating round proteins called alpha and beta tubulins, which form long strands called protofilaments. 13 of these protofilaments come together to form a single microtubule. Microtubules stretch to and from every corner of the cell, which allows them to be used like railroads for intracellular transport. For example, proteins like kinesin and dynein pick up vesicles full of proteins, lipids, or hormones and carry them on microtubules either to or from the cell membrane to specific organelles, like the Golgi apparatus. Microtubules can also resist a lot of compression force and still maintain their shape. So they're like the steel beams that support and give a building its shape. Microtubules also play a role in cell division. During cell division, there are 2 centrosomes, and each centrosome is made out of 2 centrioles. Each centriole is in turn made up of 9 sets of microtubule triplets, and each microtubule triplet is at a slight angle with respect to the next microtubule triplet. Now, during cell division, each chromosome is made up of a pair of sister chromatids which are joined together at a region called the centromere. At the centromere, there's a specific protein complex called the kinetochore. To help the 2 sister chromatids separate from each other, microtubules from the centriole polymerize in the direction of the kinetochores. It looks kind of like a fishing line being cast out to its target. And then the line gets reeled in, pulling the kinetochore and its attached sister chromatid away from its partner chromosome. Ultimately, this gives rise to two sets of single chromatid chromosomes, one for each of the daughter cells. Microtubules also help with cell movement, and that's because centrioles also form at the base of flagella and cilia. Flagella form the sperm cell's tail, which wiggles to help the sperm move. Cilia, on the other hand, are shorter than flagella, and can be non-motile or primary cilia, or they can be motile or secondary cilia. Non-motile cilia are found on photoreceptor cells in the human eye, and they help with the absorption of photons, which then the brain converts into a color image. Motile cilia, on the other hand, can be found on cells like the ones that line the fallopian tubes, which slowly sway and drag the egg cell, fertilized or not, toward the uterus. Finally, there are the intermediate filaments, which fall in between actin filaments and microtubules in terms of size. Intermediate filaments are made of interlaced threads of protein, like a rope. This makes intermediate filaments stronger than actin filaments, but also less dynamic. Intermediate filaments mostly fasten various organelles like the nucleus in place within the cell. Intermediate filaments also help anchor cells to neighboring cells through cell-to-cell junctions. They also help anchor the cells to their surrounding environment, which is the extracellular matrix. Intermediate filaments are less dynamic than actin filaments and microtubules, so they tend to degrade slowly and last longer within the cell. Now, unlike actin filaments and microtubules, intermediate filaments are made of many types of proteins depending on the cell type. They're classified by their molecular similarities into type 1, 2, 3, 4, 5, and 6. Types 1 and 2 are the keratins, and they make up the main structural component of hair, nails, and the outermost layer of the skin. Type 3 includes the most common of all intermediate filaments, vimentin. Vimentin is found in cells like smooth muscle cells, fibroblasts, and white blood cells. Another type 3 protein is desmin, and it forms the basic unit of the skeletal and cardiac muscle cells, which is the sarcomere that makes muscles contract. A third type 3 intermediate filament protein is called glial fibrillary acidic protein, and it's found in supporting cells in the central nervous system, like astrocytes. A fourth type 3 intermediate filament protein is called peripherin, and is found in neurons of the peripheral nervous system. Type 4 intermediate filaments include cinnamon, which is also called desmuslin, and this provides resistance to mechanical stress in cells. So while they're found in all cells, they're especially abundant in skeletal muscle cells. Another muscle-specific type 4 intermediate protein is syncoilin. There are also some neuron-specific type 4 filaments, like alpha-internexin, and a group simply called neurofilaments, which provide structural support for axons. Finally, there's type 5 and type 6 intermediate filaments, which are found in almost all of our cells. Type 5 intermediate filaments are lamins, which make up the nuclear lamina around the nucleus of the cell. Then there are type 6 intermediate filaments, which include nestin, which helps assemble and disassemble other intermediate filaments. Alright, as a quick recap, the cytoskeleton is formed by actin filaments, intermediate filaments, and microtubules. Actin filaments are the thinnest and help with muscle contraction, cell division, and even cell movement. Intermediate filaments fall between actin filaments and microtubules when it comes to diameter, and they provide strength to the structure of the cell. And finally, there are microtubules, which are the thickest of the three. Microtubules give cells their overall shape, form centrioles, and help in the transportation of vesicles.
Helping current and future clinicians focus, learn, retain, and thrive. Learn more